Hi, Year 7. Welcome to Lesson 2 of our topic on cells. Today we need to look at the differences between plant cells and animal cells and see whether we can label all the parts of them and see what the differences are. There are a lot of scientific terms we need to learn in this lesson. If you look at the box on the right hand side, there are 1, 2, 3, 4, 5, 6, 7, 8 new words to learn, and we're going to use those words a lot during the lesson, but it might be worthwhile for you to ask somebody in your family to quiz you to see whether you can remember what they all are. We also need to learn how to describe the functions of the components of a cell, in other words, what each part of the cell does, and we need to be able to describe the differences between plant and animal cells. And this is a picture of a cell to show all of the components that are inside a shell. You don't need to learn the names of all of those, but we will be learning some of them in this lesson. All of the components inside that cell are called organelles, and I'm going to refer to them as organelles most of the time in this lesson rather than components. Some organelles are in both animal cells and plant cells, but some organelles are only in plant cells. Here is an animal cell, and it has 4 parts which we're going to name and find out about. There's the cell membrane, which is porous and it allows molecules and chemicals to pass in and out of the cell. There's the cytoplasm, which is like a jelly where all the chemical reactions happen. There's the nucleus, which is the boss and controls the cell. And then there are some mitochondria which provide energy for the cell. So those are the parts of an animal cell. And here we have a plant cell, and there are rather more parts in a plant cell, but some of them are the same as an animal cell. Okay, the plant has a cell wall which is made of cellulose. Lots of useful things are made of cellulose, including paper. Inside the cell wall is the cell membrane, and the jelly-like substance called cytoplasm is there where the chemical reactions take place. There's the nucleus again, which controls the functions of the cell. There's a vacuole now which is filled with a liquid which helps to give the plant its shape. There are chloroplasts, which are green, which is where photosynthesis takes place. You might remember that chlorine is a green gas and chlorophyll is green, and they're called chloroplasts, and they're also green. So chloro means green. Then, as usual, there are mitochondria which provide energy for the cell. So those are all the parts of a plant cell. Here's your first quiz. I would like you to pause the video and see if you can name all of those parts of the cell. I've given you the names. See if you can match them up with where the parts are. If you're looking through the PowerPoint, you can stop at this slide because the next few clicks will give you the answers. So, answers. I hope you remembered some of them. And if you're looking at the PowerPoint, you can stay on that slide and check all of your answers. Now we're going to look at what these components in a cell do. So these are components that are in both animal and plant cells. And I've just seen that I've made a spelling mistake. They are components, not components. Okay, so the first organelle we're going to look at is the nucleus, which controls the cell, and it contains genetic information like DNA. Then there's the cell membrane, which is porous, and it regulates what goes in and what comes out of the cell. And the cytoplasm is this jelly-like substance, like cell sap, where all the reactions take place, the chemical reactions. And mitochondria are organelles that provide energy for the cell to work using respiration. By the way, mitochondria is a plural, and mitochondrion is the singular form. So those are that is what each component does in both animal and plant cells. Now we're going to look at the organelles that are only in plant cells and see what they do. So they have a cell wall which strengthens the cell, provides support. It's made of a tough fibre called cellulose, which we spoke about earlier. Then there's a vacuole which contains sap, which keeps the cell firm. That's really what holds plants upright and keeps the stems stiff. Then there are the chloroplasts, which contain a green substance called chlorophyll, and they collect energy from the sun for photosynthesis. So those are the organelles and their functions, which are only in plant cells, not animal cells. Okay, I'd like to talk to you about an analogy. An analogy is something we use quite a lot in English, but we can use it in science as well. So for this analogy, we're going to think of a bank. If you can imagine, the bank has walls with doors which control who goes in and out of the bank. It has a bank manager who's in charge of everything and tells people what to do. The bank has a floor space, which is where all the transactions take place. And the bank needs a supply of electricity for everything to work. And what I'd like you to do is to see if you can match the part of the bank with the part of the cell. So if you pause the video and try that, I'll give the answers in a second. So there are the answers, and again on the PowerPoint you can check that you can have more time to check whether they're correct. Right, this is the last slide for now. In this video we can see how slides are prepared for the microscope. Ms. Otiemi and Mr. Ero are going to prepare a plant slide and an animal cell slide, so you can watch the video and see how they do it. At the same time we can look at the process, because there are quite a lot of steps, as you can see. 9 steps. You can watch them do each of those steps for the plant cell and for the animal. So I suggest you stay on this slide and you watch the videos and see how they go through the process.
Today I'm going to show you how to prepare an onion slide. You need a sharpish knife. Obviously, health and safety, you need to make sure that if you're doing, well, you can't do it at home because you don't have a microscope. But anyway, the aim of this is to make an onion slide. So when you cut your onion, you need to be able to peel the membrane off. So I hope you can see this. Actually, I need tweezers here. There is this thin layer between each onion layer. Now that is not a very good one. Let me just put it on there so you can see it. So sometimes when you're doing this in class, you might have like 2 or 3 goes to try and remove that layer. And I'm still struggling. Oh, here we go. Now you can see it's folded up. When you're making your onion slide, you do not want it folded up at all. So I'm going to try and straighten it out. Actually in school you'll be using a scalpel rather than a knife. This is really tricky. Okay, I think I'm going to have to cut another one. Let me try and find a layer that is not on a curve. So let's try this bit here. Oh, there's one here, if you can see it. But that is way too big. Use tweezers.
That's a nice,
it's a nice large one, but we'll see. Okay. So if this happens to you in class, don't worry too much.
happens to the best of us.
It does indeed. So what you need is some iodine stain. So try and flatten it out. and remove any creases. I don't know if you can see that. Okay, and then you put 1 or 2 drops of iodine. This makes it easy to see the cells. I'm going to put 2 drops. And then you're going to get a cover slip. A cover slip is a thin piece of glass. It's very sharp. So again, when school resumes and you're doing this in class, you have to be very careful because the edges are very, very sharp. You place that on your onion membrane, and then you get a piece of tissue, which Sarah is just getting for us now.
Run!
Thank you, sir. And you mop up any excess iodine.
Gorgeous, gorgeous onion slide right there.
Yeah. Okay. Now, you put this under your microscope. So let's go back to the microscope. You switch on your microscope. You switch on the light. Make sure that the objective lens is on the lowest one, which is 4. Move the clips out of the way and put your onion slide in the middle on the stage, and then clip it down. And then you use your coarse adjuster or coarse focus to move this stage up and down so that it's into focus. It comes into focus. Right. Now, on the lowest magnification, you can't see too much detail. So I can see the cell boundaries. I don't know if you can see that.
Oh, yeah, I got a bit. Ooh, cell boundaries.
So you can see the cell walls between each of the cells.
That's quite difficult. Yeah.
Okay. And if you want to see more detail, you need to turn your objective lens to the next level. So move your coarse focus down a bit, and then refocus it. And then you can use the fine focusing to adjust it even further. Okay. I don't know whether you can see that.
Ooh, cell walls. They look good.
Okay, and then in order to use the higher magnification, move your stage down and put it on the highest magnification, which is 40. Now in school, thank you, sir. In school, what you would normally be doing is drawing the cells that you see. So what I saw under the microscope, and I'm not that brilliant at drawing, but I will try my best, is something like this. And then you just draw a few of the cells and then draw the nucleus in the middle. The cell walls here are a little bit thick. Now when you're drawing in diagrams in science, you have to make sure, you're using a pencil and you're using, stop laughing, Mr. Arrow.
Sorry.
You're using a pencil and you're drawing single lines. You don't want to sketch it because we don't do sketching in science. Okay. Let me try and see if I can focus this again. Okay, it's really hard to see. Now, once you get it in focus, you should be able to draw in more detail what you see. In order to calculate the magnification, Mr. LaPage is going to describe how you do that.
Okay. One of the questions you'll be asked in tests and exams is how to calculate the magnification of both lenses together. And you do this by multiplying the strength of each lens. So in this microscope, the eyepiece lens magnifies by 10 times. And with this biggest objective lens, it magnifies by 40 times. So if we want to calculate the total magnification, all we need to do is to multiply the two magnifications together. So the total is the 10 times 1 multiplied by the 40 times 1, so the total magnification will be 400 times. And there'll be plenty other examples of those in the worksheet.
Yay! What you've seen is a plant cell with the onion cell. So now we're going to try and have a look at an animal cell. And humans, like us, are animals. So I'm going to take some of my cells and see if I can see them under the microscope. And in order to do that, I'm going to actually take a look at my cheek cells. So this is going to be really gross. But you basically get a little cotton swab and you swab the inside of your mouth. So, oh, gorgeous, right? Oh, yeah. And then with that, then I'll put the slide on that so you can see it a bit better. And then just put some of my cells onto that slide there. And then I will throw that in the bin. And then I just get some of this methyl blue and just a drop on. And that should hopefully make it so my cells can be seen. And then with a little cover slip, just drop the cover slip on. There we go. And then with some paper towel, just dab up any of the leftover methyl blue. Oops. Okay, and then, without staining myself too much, which I've managed to do because obviously that's how it works. Then come over and I'm going to see if I can find my cheek cells under the microscope. So, I can see some of my cells.
Are you starting with the lowest magnification?
I am, yes, and then slowly but surely move higher. Obviously, it's not as easy to find animal cells as it is to find plant cells because my cheek cells are very small. Largest magnification. I will find some of my cheek cells for you and then I will upload a picture of them for you. So that is how you find your cells, your cheek cells. So as opposed to a plant cell, cheek cells, when you see it under a microscope, will be, you'll see the cell membrane and you'll see a nucleus, and that's probably all you'll see. You will not see a cell wall because animal cells do not have cell walls.
Hi, this is a reminder of the things that we need to know before we try out the quiz. We need to know all of those scientific terms in the box and the function of each component in a cell and the differences between plant and animal cells. So I'm going to give you, 15 or 20 seconds on each slide. You can also do it using the PowerPoint. So here we go. Number 1. What structure is this? Is it cytoplasm, cell wall, nucleus, or cell membrane? 2. What structure is this? Is it cytoplasm, sap vacuole, chloroplast, or nucleus? 3. What structure is this? Cytoplasm, vacuole, chloroplast, or nucleus? 4. What is the function or job of this cell organelle? Is it for respiration, photosynthesis, or does it contain genetic information, or does it control what goes in and out? Question 5. In a plant cell, do chloroplasts give shape to the cell? True or false? There's a bit of a clue in the picture. Question 6. Which part is not found in animal cells? The cell wall, the cell membrane, or mitochondria? Question number 7. Which is not part of cell theory? All living things are made from cells. Cells are always found together. Cells make up the basic unit of life. Question 8. What's this part of the cell? Is it the cell wall, the cell membrane, the sap vacuole, or cytoplasm? What's the function of the mitochondria? It lets things in and out of the cell, or it moves the cell, or it's where respiration takes place, or where photosynthesis takes place. Question 10. This is a little song. Everything's made up of cells. They contain some... I'll give you a second to think about that. And here are some word puzzles. So can you work out which part of the cell these three pictures make? Okay, so cell, mem, brain. This is a little bit more tricky. Which part of the cell is formed by these three pictures? So it's news without the S. I'm sure you got that. Nucleus. Last one. A special cell found in our body. And it's a red blood cell. Okay, well done if you got all of those right. Congratulations, you've finished the second biology lesson. Next lesson we'll be learning about specialized cells, and guess what? There is no worksheet today. We'll find out how well you've been doing when we do the end of chapter assessment on Caboodle. That ends the second lesson, and I'll be back tomorrow with lesson number 3. Bye.